This session is Envisioning a Post-Lyme Disease World. And I know it will be very relevant for all of you school nurses in New England, right? We experience way too many ticks and tick-borne illnesses in our area. So we're thrilled to have you with us. Again, the expanded bio for Stephen Rich can be found in your booklet. But for now, I'm going to turn it over to our speaker, Stephen Rich. Thank you so much for being with us virtually.
It's my pleasure, and thank you for having me.
I want to encourage people that we can still make this interactive. If you have questions, raise your hand or kind of like flag me, and I'll come up here to the laptop and let Stephen know that there's a question, and we'll kind of relay it like that. So please feel free to ask the questions when you have them, and we'll still continue to make this interactive. Okay, thank you so much. Take it away.
Thank you. Yeah, please, by all means, interrupt me. I'm a professor, so I'm used to being interrupted on occasion. Thank you very much for having me. So what I'm going to talk about today is envisioning a post-Lyme Lyme disease world. I am a professor at the University of Massachusetts. I'm also the director of the Laboratory of Medical Zoology and a group called the New England Center of Excellence in Vector-Borne Diseases, which I'll speak a little bit about at the end. But first, I have to get out just a little bit of a disclaimer document just to tell you that I am also a principal owner of MedZoo Inc., which provides tick report testing. We've been doing tick testing for several years. The operation became privatized a few years ago, and I'm a principal owner of that. I'm going to speak a little bit, just touch on it, but it's basically not relevant to most of what I'm talking about. But in the interest of the conflict resolution, I'm just making that clear. So I've left kind of a provocative title for you, envisioning the world as it would exist after Lyme disease. And if you're old enough to have watched, I can't see the room, so I don't know who I'm talking to, but if you're old enough to have watched Gilligan Island reruns on TV, then you're old enough to remember a time when there wasn't Lyme disease and there wasn't principally ticks that were associated with the disease. And in fact, it was 1975 when the first cases were reported in Old Lyme, Connecticut, where the disease takes its name from. Many of you probably know that story. You probably also know that it was not physicians or public health folks. It was a group of mothers, basically, one mother in particular, that made these alarming observations that a number of children were suffering from what seemed like juvenile rheumatoid arthritis in extraordinarily high numbers. Eventually, it was worked out that that had tick-borne etiology. Later, they figured out that it was a bacterium that was transmitted by the ticks. So in 1975, we went from basically no Lyme disease to Lyme disease being a problem. In 1991, it became common enough that it became a reportable reportable disease through the CDC. And so today, 2025, and it's been true for more than a decade, it is couple decades now, it's been the most commonly reported vector-borne disease in North America. So triple EV and West Nile virus, and you hear a little about dengue, and maybe some of you have heard of Zika or chikungunya, or all these different viruses that are associated with mosquitoes. Lyme disease, by far, accounts for much more illness than any of those. And again, I'm probably not telling you anything you don't know there. This sort of captures where we stand. So from 1991, when it became reportable, up to 2023, with a little blip around the COVID times, probably a reporting phenomenon. And the different colors basically reflect a difference in how the disease was reported over time. The title that I put forward was sort of envisioning that after this doesn't go up to 2025, but if we imagined that we took Lyme disease out of the equation, which could happen, I think it's a pretty unlikely event, but one way that we might imagine maybe not eradicating Lyme disease, but greatly reducing it would be through proliferation of vaccines that would provide protection to people. And I just want to say a couple of words about Lyme disease itself, because I'm going to focus on things that aren't Lyme disease because I think they're very important as well. So something to know about Lyme disease and all the other diseases I'm going to talk about today is they have a complex life cycle, and the ticks, that's because the ticks have a complex life cycle. They feed on multiple different hosts in the course of their life. Most ticks, in many instances of different diseases, are born without infection, so they have to acquire infection through a blood meal that they take on a organism, wild animal. And typically, we would say that these are zoonoses, meaning that they're diseases that are actually diseases transmitted from tick to animal in the wild and maintained naturally that way. And they're really only a problem to us when they spill over as a manifestation of just an abundance of the ticks that break that cycle and feed on us. So that's a fundamental thing for us to understand about what makes Lyme disease and everything else I'm going to talk about today different than other anthrapanoses or diseases that get transmitted person to person. And one of the ways you can think about this, and we heard a lot about this during COVID, is that you get no benefit of herd immunity in the case where you do something like vaccinating against Lyme disease. It's one of the reasons that seeing our way out of Lyme disease through vaccination seems relatively unlikely, but for purposes of our little thought experiment, we can imagine that there will be vaccines on the market very soon. There are some in trial. Some of you may know this. There's been vaccines in the past that have been taken off the market, but none of them will give benefit or will have the benefit of herd immunity because by vaccinating people, we're not doing anything to reduce the source of infection. So whereas in COVID vaccination, if we are vaccinated against COVID, we become no longer a source to infection to others. Not true in the case of Lyme disease. So if we were to eradicate Lyme disease, then we'd have to continuously to vaccinate people to keep it away. So my reason for presenting this thought experiment of imagining a world after Lyme disease is to get you to think more about the biology that's involved with ticks and tick-borne diseases and not hung up so much on Lyme disease per se, which probably a number of us know a lot about. And so the first thing I would say that would be true if we got rid of Lyme disease tomorrow is that we most likely would not get rid of ticks. There's no effort underway and probably no means at our disposal presently to get rid of all the ticks. I alluded to the fact that ticks were not really a big problem 50 years ago, and that's largely because of changes in the landscape, changes in the way that we've maintained our landscape specifically. And probably one of the principal reasons for the proliferation of ticks is that we've proliferated our landscapes in a way that there are an abundance of white-tailed deer that hasn't been known in any historical times. And since all of the ticks that I'm going to talk about today feed on white-tailed deer, and two of them rely almost exclusively on white-tailed deer as their reproductive hosts, deer are a fundamental problem here. So here's a sentence just to set a couple of definitions for us going forward. So I said ticks are vectors of disease caused by pathogens. And of course, you know that vectors include things like mosquitoes and fleas. Ticks are in that category as well. The diseases include things like yellow fever and malaria and triple EV and West Nile virus, but also Lyme disease. And they're all transmitted, those diseases are all caused by different pathogens. So we make an important distinction between a disease and the pathogen or the microbe that causes that disease. So I use examples here, viruses and parasites. And I've highlighted in red the creature that we're sort of eliminating today for purposes of our conversation, just to remind us that ticks are a bit extraordinary in a number of ways for mosquitoes and fleas. First of all, they weren't known to be, before 1975, they weren't known to be human risk factors or public health concerns. They were largely agricultural concerns. So there was concern about diseases transmitted to cattle and in some cases maybe to poultry. But for the most part, and especially in North America, they weren't considered to be associated with public health concerns. So that puts them automatically in a different category. It means that we didn't have much of the 20th century to build up things like mosquito control districts where we could take widespread measures to try to control mosquitoes. but there are also just some basic aspects of tick biology that makes them not so amenable to the control measures that we use against mosquitoes. The first bullet point here I already made that ticks are, for most of human history, have been thought of as agricultural concerns. I mentioned when I showed the life cycle slide that most of the diseases are enzootic, which makes them different than things like dengue fever or malaria because they're not maintained in our populations. They're maintained in populations of wild animals that the ticks feed on. And then, of course, ticks have a very different distribution than mosquitoes. So you may know that mosquitoes breed in water, but they breed and they spend their early part of their life in water. This makes them susceptible to kinds of control that we don't have for ticks. Ticks are much more patchy in their distribution, making them harder to identify and kill individually, and making it a real challenge, as I mentioned before, to think about trying to eradicate ticks themselves. So all of this is to say that while ticks weren't a problem for our parents or our grandparents' generation, they are a problem for us, and they're going to be a problem for some time, even if we're able to develop vaccines against things like Lyme disease that reduce or limit the transmission of those pathogens. So what I want to spend some time talking about is that biology of the ticks, because it will also inform, although I won't speak much about Lyme disease directly, it will help to inform some of the things that you may have heard about Lyme disease, and that now you'll understand maybe some of the biology or behind why some of the things are asserted about ticks and Lyme disease. So this is hopefully it's big in the room there. I'm sorry I can't be there, but these are the tail of the tapes with respect to human biting ticks here in New England. There are no less than 3 species of human biting ticks, and they are the deer tick, the dog tick, and the Lone Star tick. And you notice that in each set of images, there are 4 separate ticks. That's because there are 4 separate life cycles. There's the larval tick, that little tiniest one, in each case in the lower left. There's the nymphal tick, which is sort of a teenage tick, and then the adult males and females, which are sexually dimorphic, as you can see. And the females in many species are larger than the males because they're the ones that are going to be producing the eggs. And it's important to know that a basic aspect of tick biology is they take exactly 1 blood meal in each of those stages. So each tick on this slide will have, if it runs the course of its life, will have taken 4 blood meals. 4 times it will have fed on a host that it derives a full blood meal, and then it uses that blood meal in the case of the larval and nymphal stages to advance to the next stage. In the case of the adults, they use their blood meal to convert to eggs. So they use that specifically to lay the thousands of eggs. And I'm going to show you a picture of that in a moment. And I should point out that male ticks, like many male mosquitoes, don't feed as adults. And so they spend their time looking for females. And the females are the ones that are predominant blood feeders in that stage. So just a notion of the distribution. So you'll notice in all of these slides that New England is in the geographic distribution of things like the black-legged tick, which is also known as the deer tick, which does transmit Lyme disease, but as you'll see in a moment, it transmits a host of other pathogens as well. The American dog tick, located throughout the east, it's actually west of the Rockies as well, although we think it's a separate subspecies, but certainly throughout New England. And then a relative newcomer, which is the Lone Star tick, which, again, not known in New Hampshire, probably not known outside the coastal areas. Here in Massachusetts, we see it in Cape Cod. We see it in coastal Connecticut and Rhode Island. It's one of the predominant ticks on Long Island, but it's really just creeping northward into the inland interior of New England. It's a third tick. It's probably here to stay. It's probably going to increase in numbers before it decreases. People ask, does that have anything to do with global warming? Probably not so much. Global warming is probably facilitating its movement a little bit. But as I said before, by and large, the real reason that these ticks are expanding their ranges and increasing in number is that there are more deer, and deer are basically breeding grounds for especially the Lone Star tick and the black-legged ticks. So those are 3 human-biting ticks that are present here in New England, and each of them really is expanding their range. Black-legged ticks have been throughout New England for a decade and a half now, and creeping into southern Quebec and Ontario as well. Let me dispute. One of the things I want to do is dismiss a couple of myths about ticks. So one of those myths can be shown by this little video, which I'll show you. It has tick activity. So you see a young lady walking down a trail, and you see what ticks do. And I emphasize that they don't jump, they don't fly, they don't fall from trees. All they do is what this little creature is doing here is they crawl out to the end of a branch, and then they do something called questing. So there's a female tick questing. She sticks her legs out and she waits for something to grab onto, in this case, this passerby. And then there's a length of time that the tick, will spend crawling around on the surface before she finds a suitable place to take a blood meal. It's not altogether clear what they're looking for. They do seem to like things like waistbands of our clothing, where there's sort of a tight fitting of the clothing to our skin. They also like, I say parts of our body that form a Y, so under the arms and the groin, along the hairline, and they'll seek those areas out and then they'll attach. And here's a crucial thing to know is, unlike mosquitoes, which you may know, mosquitoes feed in 30 to 60 seconds and they're done, they fly away. Ticks can remain attached and feeding for the better part of a week in the case of adults, even up to 10 days. So they'll take a gigantic blood meal that's several times their own body weight, and then they'll use that as the energy source that will take them to the next stage. I think I may have skipped over saying that ticks only feed on blood. They don't feed on anything else. So there's some evidence that maybe some ticks will occasionally drink a little bit of water off a leaf, but for the most part, everything that goes into a tick comes from the blood meal that it takes, and they take exactly one of those blood meals in each life stage. So this is just another way of showing that life cycle I showed you before, the 4 different pictures of the different life stages. Here's the eggs and the larvae and the nymph, and I've shown just an adult female here. And I want to show you a couple variations that ticks have on those life cycles. There's a female tick laying her clutch of 2,000 to 3,000 eggs to give you some idea of the problem here. So if one gravid female can lay 3,000 eggs, and one white-tailed deer can feed hundreds to thousands of adult female ticks, then you can imagine how the numbers can increase relatively quickly when there's just a small number of white-tailed deer that get infected with the females. So in terms of life cycle, we say that ticks have we categorize them by the number of hosts that they have. So you'll notice that all these slides have agricultural hosts, and that's again because the ticks are predominantly feeding on either domesticated companions or wild animals. They don't do particularly well in our houses. So if a tick gets on you or your cat or your dog, it will fall off and maybe try to get on to another host at a later time. But for the most part, our houses are too warm or too dry, or in some cases too cold for ticks to survive very long. So they really do live in the wild and depend on these wild animals to feed. A one-host tick is a tick that gets on the host and then spends its whole life on that host. And so they pose less public health risk to us because they're basically doing everything on that one host and don't fall off and get onto us. When you get to two-host ticks, where a tick will feed on one species and then later in a later stage feed on another, that's when we start to see the pathogens be bridged from the wild animals into us. Lyme disease is actually a three-host tick, is transmitted by a three-host tick. So the black-legged tick has 3 hosts. And here's the fundamental problem is that the ticks as juveniles are feeding on mice, which are the predominant source of infection with the pathogens, Borrelia burgdorferi, for example. They pick up infection and then they carry them throughout their lifespan. And if we happen to interrupt and jump in the way of a tick that was destined to be on a white-tailed deer, that's when we get exposed. So not only are there ticks in addition to the black-legged tick, so the black-legged tick is the one on the far left there, that's associated with Lyme disease, but they're also each associated with a different set of pathogens. So Lyme borreliosis is the fancy name for Lyme disease that's at the top of the list in the black-legged tick column. But there are other borrelias like Borrelia miyamotoi, which is a relapsing fever spirochete. And I'll show you some of the percentages of infection with these different pathogens in a moment. There's also things like Babesia, and I'm going to go into a little bit more detail on each of those in future slides, so I won't spend too much time on those. Dog ticks are very uncommonly infected, but when they are infected, they can be infected with some pretty dangerous pathogens. So they carry things like tularemia, Francisella tularensis, which can cause a very debilitating skin lesion, and in some cases, a fulminating infection that can be fatal. And then, of course, Rocky Mountain spotted fever, though both of these are very, very rare, meaning that they occur in probably less than a tenth of a percent of the ticks. In fact, we rarely see these pathogens in dog ticks. So while dog ticks are a nuisance and often abundant, especially in June and July in New England, they tend not to transmit many pathogens. And then on the far right is the one that I told you that's the newest comer, which is the Lone Star tick. It also has a set of pathogens associated with it. They also are relatively rare. Things like ehrlichiosis, transmitted by a pathogen called Ehrlichia. Um, it's quite uncommon. In fact, we've yet to see it in the New England ticks for the most part that have been established that are recently arrived here. So, for example, we see 20 to 40 Lone Star ticks in a sampling we do on Cape Cod, in Cape Cod, Massachusetts, and none of them yet have been infected with Ehrlichia. And that's probably because the infection and the migration of the infection and the migration of the ticks is not completely coincident. At the bottom here, I put the oddest one of all, which again is probably something that you've heard of before. The formal term for it is alpha-gal syndrome or alpha-galactose syndrome, or commonly referred to as a red meat allergy. And this is unusual in that, and I've highlighted it because unlike the other diseases that appear here, or the other infections that appear here, it's not associated with a pathogen, not a virus, not a bacterium, not a parasite, but rather it's an illness, an allergy that develops after the bite of one of these ticks. And the idea is that there are sugars on the surface of red meats. There are sugars in the tick that mimic that. And so one becomes sensitized to the red meat, to the sugar that's like the red meat sugar when after the bite of a Lone Star tick, and then later develops an allergy to red meat. I'm going to speak a little bit more about those risks and about what's known when I come to the section where I talk about risks. So I'll set that aside. And again, if there are questions and you want to pass them along to Brianna, I'm happy to answer now or wait to the end. So I showed you the picture of nymphs before, so or the life stages. Most of the diseases that are transmitted, including Lyme disease, are transmitted by that tick that's in the red circle there. That's the nymphal tick. It's not to say that adult ticks aren't infected. Larval ticks, it turns out, are not infected, but adult ticks are also infected, but they transmit less disease because they're bigger and they tend to go to get noticed before they feed for a long period of time. Nymphs are just the right mix of they have infection and they're small enough that they'll feed long enough in the hairline or again along a waistband or that kind of thing. They'll feed long enough that they can actually transmit pathogens. Yeah, so that's why nymphal ticks are really the biggest risk factor, especially for Lyme disease, because they're larger in numbers. There are more nymphal ticks than there are adults by definition. They're smaller in size, and they're abundant in summer when people are active. So these are ticks that are active basically from the end of May into like just about now, just about the first week of August, they start waning off. People don't always realize this, but we're actually coming into a doldrums period for tick activity. So August and September, in terms of the warmer months, are some of the best times to avoid activity of ticks. It's not to say there aren't any. It's just there are much less than there were in May, June, and July. So let me just go through a couple of those different pathogens and give a little bit of details of them. So one of them is a relapsing fever like spirochete that's caused by a bacterium called Borrelia miyamotoi. It has a worldwide distribution. It was first identified in Japan. It's in about 1% of the black-legged ticks that we see here in New England or across the United States, actually. Um, and it's also transmitted by the Western version of the black-legged tick called the Western black-legged tick. It causes a disease quite distinct from Lyme disease because Lyme disease is not a bloodborne infection. It has a transient phase where the pathogen moves through the blood and then sequesters in tissues. This thing is actually bloodborne. It stays in the blood and it transmits, or sorry, it circulates through the blood and then undergoes antigenic variation to avoid the immune response. It's associated with something called relapsing fever, which is, as the name would imply, sort of a recurrent fever that's associated with those different antigenic profiles of the bacterium itself. Because it's relatively rare, it tends to go to go missed and often undiagnosed, but it is something that occurs in about 1% of the ticks. I lumped these two together, ehrlichiosis and anaplasmosis, because at one time, microbiologists and entomologists like to change the names of things. But one time they were all the same thing, and now they've sort of been split between ehrlichiosis and anaplasmosis. And there's a couple different varieties of them. So ehrlichias are transmitted by black-legged ticks and dog ticks, and anaplasma is transmitted by black-legged ticks almost exclusively. These are also bloodborne infections. They're intracellular infections, which is also different than Lyme disease, meaning that they actually get into, in the case of anaplasma, into neutrophils. So they get into granulocytic cells that are meant to be part of our immune system. So it makes them particularly nefarious um, and and somewhat more widespread than Borrelia miyamotoi. Again, I'll show you numbers in a moment, but not quite as common as Lyme disease. And then the other one on our list, which is not a bacterium, so everything I've spoken about heretofore is a bacterial pathogen, but Babesia is actually a parasite that most closely resembles malaria. It's transmitted by the same ticks that transmit Lyme disease. So again, if we got rid of Lyme disease, we'd still have anaplasma, we'd still have Babesia, and you'll see the numbers of those in a moment. There's a couple of different species associated with transmission in the black-legged ticks and disease in humans. Um, Occasionally, it does make its way into the blood banks, and it gets transmitted through transfusions. In fact, some of the fatalities that have come about in recent years are attributable to contamination in blood banks. And then the last one that I'll talk about today among these diseases that are other than Lyme disease is Powassan virus. Powassan virus is an interesting one. It's a virus, so it makes it different than the bacteria or the parasites. There are actually two different types. One has been around for a long time. Type 1, we've known about it. It's typically associated with something called a woodchuck tick. And if you're not a woodchuck, you probably don't often see woodchuck ticks because woodchuck ticks feed in woodchuck burrows. But recently, 20 years ago now, that it was discovered that there's a separate type of Powassan virus that's transmitted by the black-legged ticks, by the deer ticks. And so that's become of principal concern as well. Most of the cases, as indicated here in the northern US, in extreme cases, it causes encephalitis. So for many years, we sort of bemoaned the fact that every instance, every outbreak, as it were, of Powassan virus would make its way into the Boston Globe or the New York Times and be a pretty high-profile case because encephalitis is life-threatening and the survivalship in Powassan encephalitis is relatively low. But what we've shown by testing human biting ticks is that many, many people get bitten by ticks that are positive that they're infected with Powassan virus and show no symptoms whatsoever. And so probably those encephalitic cases, albeit very unfortunate, very tragic, they're probably just the tip of the iceberg of exposures. And many of us probably get exposed to Powassan virus with no ill effects. This would be analogous to West Nile virus, which is a mosquito-borne virus that's closely related to Powassan, in that many people get bitten by mosquitoes that are infected with West Nile virus and maybe even seroconvert, so have antibodies against West Nile, but have don't develop the symptoms of of advanced disease. So Poisson's probably in that category. There's still much to be understood, um, not to dismiss it entirely, but I think sometimes because of the nature of the severity of those cases that get reported, it may be out of balance in terms of the degree of concern about this one. So there's a map that sort of shows the distribution. So there's a lot of purple there because that's Lyme disease. I didn't take that out, but you see the distribution of these different pathogens, and you'll notice that things like Anaplasma and Babesia are throughout New England. The Rocky Mountain spotted fevers tend to be further south, associated with, in many cases, something called a brown dog tick that we don't have here, fortunately, in Massachusetts. Um, but you see that these diseases are widespread. And so imagine this image without the purple. And that's the reason I say if we tried to think about what the world looks like after Lyme disease, well, we would still have a major tick problem or multiple tick problems on our hands, given the abundance of these other pathogens. Oh, so here I've got just a reminder. I told you about these pathogens and that those, oh, sorry, those are repeats. Um, Yeah, I'm sorry. I thought I had a slide out of order there. So what I want to talk about just for a moment is how we go about assessing the risk of tick-borne diseases. I think sometimes this is a little bit confusing in that we know that every tick-borne disease starts with a tick bite. It's necessary that the tick bite and that it bite to repletion, basically, to transmit the pathogens that are inside it. Um, Not every tick is infected, and so in assessing risk, it's important to take consideration of the fact that some ticks are not infected. And the risk of human infection increases with feeding time. So even if a tick is infected, it has to be on for an appreciable length of time in order to transmit the pathogens. You've probably heard this before. You probably heard this public health message from your state Department of Health, or maybe a local board of health, or maybe you've read it in a journal. And I want to break that down a little bit and tell you the biology behind that. Something that happens quite commonly. So today it's great to be talking to professionals. Very often I'm out talking just to the general public, trying to explain this. And I learned early on that if I make a statement like, I say that ticks have to feed 48 hours to transmit Lyme disease on average. Invariably, a hand will shoot up and someone will say, I had a tick on me for 6 hours and I got Lyme disease. You're all wet. And what I remind them is that this is just the best science, and we're talking about a distribution, the average feeding rates and transmissions. And so you, of course, will understand that biological reality that it is possible for things to transmit in less than 48 hours or more than 48 hours. But what we're talking about is just the average rate at which the pathogens move from ticks into their hosts. And I'm going to explain the biology of that in a moment. So the way I like to think about risk, or I like to think about ways of mitigating the risk, is in terms of the timeline of a tick bite. There are things that you can do prior to being bitten by a tick to prevent the tick bite from occurring altogether. And that's one that we tend to focus on, so I don't want to gloss over it. And there are some very good things you can do. There are some very excellent ways to keep ticks off you. But I don't want to lose sight of the other opportunities we have to mitigate the risk of disease. But let's start by talking about the tick bite diseases. The first thing is to just know the life cycles. So these are life cycles. These are data. These actually are for 2017. It looks basically the same every year. The amplitude might increase or decrease a little bit. People call me every spring and say, is it going to be a big tick year or is it going to be a small tick year? My response is always the same. It's going to be a tick year. We see these patterns year after year after year with the deer ticks, which are shown in the baby blue here, sort of rising up. And this was a little bit earlier, sort of mid-April, and then descending into the months of August, as I mentioned to you before, this doldrum period. And then in the case of the deer ticks, there's actually a second peak that occurs in the fall, starting around mid-October and then ending basically when the snow falls. In the background, you see the green and yellow peaks that tell you the activity periods of the lone star ticks and the dog ticks. And you notice that those are basically summer ticks. They don't have a second peak. I want to show you a slide that looks similar to this, but you'll note that there's an important difference between this. This is seasonality of of deer ticks exclusively. And so those two peaks that we saw before, the one in the spring and the one in the fall, shown by the Julian calendar week on the bottom, those are adults, the brown bars, and then the nymphs are the blue bars. And these are human biting cases. So you'll note that more people are bitten by adult ticks than nymphal ticks, even though nymphal ticks are the predominant vectors associated with transmission of disease. And that's because they feed for a longer period of time. So while more people get fed on by adult ticks, they probably don't feed as long. Those ticks probably don't feed as long. So, in terms of your understanding of how you can keep yourself, your family, your patients, your friends uh, um, free from ticks, it's important to know this seasonality, to know when we have to be most on our guard. And you'll notice that that's in these periods, March through basically the end of July, and then again in the fall. So, what can you do? Well, you can consider using a repellent. The two that we recommend are DEET or picaridin. So DEET has been around for coming up on 75 years now. It was developed shortly after World War II. It was used as a mosquito repellent. It was initially thought that it could be used as a pesticide. Turns out it doesn't kill mosquitoes. What it does is it repels mosquitoes. It also repels ticks. So the application is on your skin. And we usually recommend a product that's in the neighborhood of 30%. More than 30%, you're probably not getting any benefit of the additional percentage. Lower than it might not be quite as effective. Many of you will know that one of the downsides of DEET is that it's a plasticizer, so it will melt plastics and rubber materials like in your sneakers. That's not not very favorable. And so we often recommend as an alternative picaridin, which is as effective as a repellent. You notice I don't have many other products here, and that's because there's lots of things, even my wife is a strong proponent of Skin So Soft and other such products, natural products, um, essential oils. All I can tell you is that in the laboratory and under rigorous testing, most of those compounds don't seem to have any effectiveness against ticks. Many of them don't have much efficacy against mosquitoes either. So while they may smell better or give us some security that they're natural as opposed to synthetic compounds, they probably aren't protecting very well against ticks or mosquitoes. So that's the first measure of personal protection we can take is repellents on the skin. You can also do things if you don't want to put those things on your body, which many of us don't. You can just think about your choice of clothing. So you can wear long pants, and you can do this clever little trick of tucking the pant legs into the socks. Um, it's an odd fashion statement, but the reason that it works is remember, ticks don't fall, jump, or fly. So they crawl up from the ground upward. And so if a tick gets on you low in your body, like on your feet or on your ankles, and it crawls up your pant leg, if it crawls up your sock on the outside because your pants are tucked in, it has a better likelihood of being detected by you, you see it and knock it off or pick it off, or just getting brushed off because they will get brushed off as you continue going about your day. So that's one way to reduce ticks. And we say light colored clothing or light colored socks, not because ticks have a preference for color, but because the dark ticks are easier to see against the light colored fabrics. And of course, checking for ticks is fundamentally important. No matter what you do, whether you use a repellent or wear the right clothing, we should always check your clothing, check your body at the end of the day. If you've been out gardening, you've been out hiking, the first thing you might want to do is jump in the shower anyway, and that's a good time to check your body and remove ticks. Um, and do it do it with other family members too. Kids, it's easier depending on how old your kids are. My kids are teenagers now, so they're not going to let me check them for ticks anymore. But the little kids, certainly you can be checking, along the hairline and encouraging them to check themselves. It's a little uncomfortable, but one place to check for ticks is in the bathroom, especially when you're sitting on the commode. It's like you're able to visualize all the parts of your body and look around the backs of your legs and that kind of thing because they don't get on the palm of your hand. They get in the parts of your body that form a Y. And so tick checks are fundamentally important. Another personal protection measure, which is very different than repellents, is you can use an acaricide, or in this case, a pesticide, to treat your clothing. So permethrin is a product. There's many different brands. This is just one of the brands that I like this one because it's in a pump spray, and so it's a little bit better. There are others that are in aerosol cans, but I find very often that the propellant wears out before you get all of the product. And permethrin is used to treat fabrics. So the molecules adhere to the fabric. When the ticks crawl on the fabric, they absorb the pesticide through their feet, their legs, and then they die. And it happens relatively quickly with black-legged ticks. These have very good safety profiles. They're something like 2,000 times more toxic to ticks than they are to people. So very low toxicity in humans. Not really recommended for babies or children under age of 1. There is some toxicity of permethrin in its liquid form to cats, so you'd want to be careful about applying or having it available if you have a pet in the household. But for the most part, it's used to treat clothing. You spray it on the clothing, you allow the clothing to dry, and then there's a protective layer that will last for a couple of washes. There are also companies that sell treated permethrin treated fabrics or other treated fabrics that so that you don't have to do the treating. It's a little bit more expensive, but the claims are a little bit higher in terms of the resilience in washes, the number of washes they'll persist. People ask about products like this and they say, do they kill pollinators? And the answer is yes. So there's nothing specific about permethrin that it kills only ticks and not butterflies or not bees. But generally, because it's on your clothing, it's not regarded as a big environmental um, it doesn't have so much environmental impact because it's killing ticks and mosquitoes or bees that land on you and not just widespread in the environment. That's not true for other things that are spread. So there are perimeter sprays. In fact, occasionally permethrin or bifenthrin are used as these are both synthetic pyrethroids are used to treat backyards. The idea is to basically develop a barrier. This is an aerial shot of my house. You see my house in the driveway of my neighbor's house, and the idea is to maybe I can do a pointer here. I don't know if you can see my pointer, but the idea is to create sort of a barrier around the house using the bifenthrin, spraying the leaf litter to kill ticks in that barrier and slow the progression into people's backyards. Um, this is more environmentally impactful than wearing clothing that has treatment on it. It is done in a way that it's meant to be judiciously applied to leaf litter and not to flowers or places where butterflies or pollinators are likely to live. But it's not to say that it's not without consequence on other insect fauna. So it will kill beetles, it will kill ants. Um, yeah, so it has many downsides. Right now, our center is doing a study. We're in our third year actually of it's called Project Itch, where the Project Itch ITCH stands for Is Tick Control Helping? There are so many households that are utilizing these backyard sprays. We really wanted to be able to give people an answer about whether it's effective or not. And the results of that will come out sometime in the next couple of months. We're working with residents throughout New England. And then the next stage will actually be to look at the consequences of where it is being effective. Is it also having off-target effects against different insect species? I said this before, so natural products, many different natural products on the market. For the most part, they don't repel ticks, they don't kill ticks, and they may not, in some cases, not be safe. In some cases, they just because they're natural. I mean, poison ivy is natural, but not particularly. And I'm not saying any of these have the effects of poison ivy, but you understand the point. So there's often a notion that we're going to do ourselves a favor by using some of these naturally occurring products. I'll tell you my mode. I don't use I use repellents to keep mosquitoes off, especially nuisance mosquitoes. But for ticks, I rely on the clothing I wear, untreated, and doing tick checks. And I've been at this for 40 years, and I've never had a Lyme titer. I've never had Lyme disease. I've been bitten by multiple ticks. I usually find them before they feed very long. And yeah, just my personal testimony. So in terms of just controlling ticks in general, we can do things like host management. So there's been some thought of trying to reduce the number of ticks that feed on white-tailed deer. Many people will suggest that maybe we should reduce the number of deer. And unfortunately, that's not going to ever work to an effect that it's going to reduce the number of ticks. You'd have to harvest so many deer that it would be, it's just not practical. I show a picture of a little island here. The picture on the right is Monhegan Island, where I worked as a graduate student, and they did eradicate their deer population. But that island is a mile long by a half mile wide. And so they were able to eliminate all of the 20 or 25 deer that they have on that island and get rid of their Lyme disease problem, or their tick problem, rather. Um, we're not going to be able to eradicate deer. We are looking, our center is also doing work to look at ways that we may be able to reduce the abundance of ticks on deer. So you know that there are products that you can feed your pets, your dogs and your cats, that will kill ticks. We're looking at some of those oral treatments and ways to deliver them as ways that might reduce the burden of ticks on the backs of deer, but we're still a couple of years away from actually having an application there. Ticks are seasonal, but just a word about ticks in winter. Very often you hear people say, oh, ticks are year round now. The truth is, I showed you those slides before showing you the seasonality. They're abundant in certain times of the year. That's not to say that you may not see a tick in New Hampshire or Vermont, northern Vermont, or northern Maine or western Massachusetts in January. You know, we get those periods when it freakishly sort of warms up to 60 degrees in the month of January, and ticks will become active. But they're just not in the same abundances that you'd see in the other times of the year. So they will come out of, they don't, black-legged ticks don't hibernate or estivate, but they will become active. And as far as the notion of what snow or bad or cold weather does to ticks, the snow actually provides a bit of insulation over top of the leaf litter and allows them to persist through that cold winter and then emerge in the fall. So the notion that a cold winter is going to eradicate ticks is not true. They've been surviving cold winters for millennia. So that's sort of an overview of things that you can do to try to keep ticks off you, things you can do on your personal, your body and your personal being, and as well as things you can do in your yard, and then things you can think about to maybe keep ticks lower in the environment. But that's not the end of the story, because we shouldn't think of a tick bite as an event that's going to lead to a necessary outcome. There are things that you can still do to greatly reduce, greatly mitigate the risk of infection after that tick bite. And one of the big ones is just pull that tick off. If a tick gets on your body, if it's not attached, then it poses really no risk at all. If it is attached, you need to pull it off as quickly as you can, or if it's attached to a patient or a subject that you're working with, then you need to get it off as quickly as possible. And there's really nothing complicated about it. So there's one little fancy forceps down there, and there's some other products that I've seen online. Some of them work okay. The idea of the thing on the lower left is called a tick twister, and the idea is you're supposed to corkscrew the tick out because, well, it's just not consistent with their biology. The ticks are not corkscrews. They're not screwed in. They don't need to be unscrewed to pull them out. And anything that you're doing to unscrew them might just be sort of making more of a mess of the situation. What you do want is to get a tweezer or a forceps, and pull them out as closely as you can to the skin, and try to get the whole tick out. And I'm going to speak to that point in a moment. But all these little devices are, a bit of overkill and maybe save money. There's a picture. So you just grip it close with a fine pair of forceps. And if you're out on a trail and you didn't bring your forceps with you, or you didn't bring your tweezers with you, and you can't do this little move, then do the best you can to try to grab it with your fingernail and pull it off. There's a bunch of different myths, and I want to say that none of these would we recommend. Don't use a burnt match and hold it to the tick and expect that she's going to back off. Don't put Vaseline on it thinking that you're going to suffocate it. They have extraordinarily low metabolism. They breathe very, very slowly. They don't have lungs, actually. They breathe passively. And so putting Vaseline just means that the tick will live for a long time. And the whole time that it's alive, it can transmit pathogens. The same is true for nail polish and Another one that we hear very often is people say you put a little bit of soap in a cotton ball and rub it. I think you have to do it in clockwise fashion or something. Again, this sort of seems to be tied to that notion of corkscrewing. Um, if you've done that and it works, good. I'm great, and I'm glad for you. We don't know why it works. It's very likely that if that worked, that tick was going to fall off anyway, because eventually they do fall off on their own. We don't really recommend anything that slows the progression of getting that tick off you, and certainly not burning yourself. So anything that involves keeping the tick on for any longer than it has to be is detrimental, because one thing we know is that ticks, the longer a tick stays on you, and the longer that tick is on you and remains alive, the higher the likelihood that it transmits a pathogen out of that tick and into you. There's a couple of other myths here. One is that if you don't remove it right, that maybe you're going to squeeze the tick with your fingers and it's going to regurgitate. Well, it's not physiologically possible for ticks to regurgitate. They have a pharyngeal block, and anyway, the stuff that's inside them isn't in the midgut, which is the stuff that's connected to that tube. It's the germs are actually in the salivary glands. I'll show you an image of that in a moment. And crushing ticks may expose you to pathogens. Just not true. When you crush a tick, when you kill a tick, because ticks are not syringes, ticks are biological entities, and they actually produce an environment that the bacteria can thrive in. And outside of that environment, including when the tick is dead, the bacteria don't live particularly well, or they don't live particularly long. So pulling ticks off, even if it means accidentally breaking a little piece, the other notion is that maybe people say, well, what if I leave the head behind? And I'll very often I'll point out that ticks don't have heads. So anatomically, they lack heads. They do have mouth parts, and the mouth parts may break off and remain in the skin. But once they're disconnected from the tick, they pose no risk of infection. Any risk of infection came prior to the separation of that tick from its mouth parts. And any parts that are left behind, they'll just work themselves out over the course of the day as a splinter of wood would. So, long story short, if you get bit by a tick, get it off as quickly as you can. You greatly reduce the risk. And if you fail to find a tick or fail to find a tick early enough, so you enter into this post-tick bite phase where the tick is fed on you and finished and left your body, or finished very closely and you find it very late stages, there are still things you can do to mitigate risk. And the reason I think that we often get confused about this is we don't stop to remember how we measure risk. So I use the example here of shark as an example of how we talk about risk. The hazard on the left is the shark in the water. So the green image of the individual standing on the beach looking out at the shark. The shark is the hazard. The risk is on the right, and that's the incidence with which the people in the water are attacked by the sharks. That's the risk as we measure it. That's why when we measure things like the incidence of Lyme disease, we talk about the number of cases per 100,000. It's the outcomes. It's the outcomes of a hazard being exposed. And so it's that middle piece, the exposure, that we often lose sight of. The exposure is the fundamentally important measurement piece in terms of understanding risk, as it were. So if we think about the same slide, sorry about that. If you think about exposure in terms of tick bites, we know that there are three fundamentally important things to know about a tick bite. First, we have to know the tick species. So I hopefully have impressed upon you that there are different tick species and different pathogens transmitted by different tick species. Not all species have the same hazards. Some have very low levels of hazards. The second is to know something about the infection status of that particular tick. So things like Borrelia burgdorferi, Lyme disease spirochetes, are present in black-legged ticks, but they're really only present in about 50% of the adult ticks and maybe 30% of the nymphal ticks. So not all ticks are infected. And you have a better than 50% chance on average that the tick that bites you is not infected. So understanding something about the infections of a particular tick will shed some light on the risk that it poses. And then that last one, which I've sort of been dancing around, is this notion of the duration of feeding. I mentioned that many of you have probably heard this before, that ticks have to be on you for an appreciable length of time before they transmit pathogens. That's a biological reality that reflects the way that pathogens move into ticks and out of ticks. We say for things like Lyme disease, the ticks have to be on anywhere from 24 to 48 hours, probably 48 hours being the average, and that the infection status is sort of relatively low. So all of these things are ways of measuring risk in a very real way that goes much further than just looking at the case reports and the number of people. Let me show you an example of how we can sometimes be misled by just simple data. So I showed you this slide before. This is an accurate picture showing the distribution of black-legged ticks or deer ticks, Ixodes scapularis, that transmit Lyme disease. So where the yellow is present, that's where black-legged ticks are found. And what I'm going to do is I'm going to superimpose the 20-year case reports for Lyme disease as dots. And so look what happens there. So there's really two foci. There's one here in the Northeast throughout New England. Massachusetts, you'll notice, has sort of looks like a blank spot there. It's because for several years, Massachusetts didn't choose to report Lyme disease. And so it leaves it blank there. But there's the Northeast foci and then a focus in the upper Midwest. And then throughout the Northeast, just to go backward, or Southeast rather, where ticks are abundant, there just isn't Lyme disease. And the reason for that is that the pathogens are absent and the ticks don't feed on people by and large. I'm not saying no ticks in the Southeast, but the black-legged ticks, the Ixodes scapularis ticks, a very specific species of tick, tend not to feed on people. They also tend not to feed on the hosts that transmit the pathogen. And they don't have a 2-year life cycle, which is fundamentally important for maintaining disease transmission. So this is an example of how one would think that maybe just the presence of the ticks in the Southeast means there's a risk of Lyme disease, but the evidence is that the risk of Lyme disease in the Southeast is relatively low. And until we go through and sort of break down that biology, in some cases tick by tick, we can be very much misled about inferring risk. So I mentioned alpha-gal syndrome. This is very much alpha-gal syndrome, AGS, the red meat allergy, is very much at the stage that I think Lyme disease was 35, 40 years ago in terms of its diagnosis. We know that there's a hazard. We know that there's some association between a particular tick species, the Lone Star tick shown there on the left, and that there is the risk of acquiring this red meat allergy. We'll say the red meat allergy, the alpha-gal syndrome, has a range of different manifestations. You may know people that are suffering from it, from maybe stomach upset after eating a cheeseburger kind of thing, to some cases anaphylaxis from exposure to shampoos or different byproducts, animal byproducts in cosmetics. So there's a wide range of responses to these allergens, and there's much yet to be understood about exactly where that exposure is and how we understand it. We don't know anything about whether the duration of feeding presupposes people to getting developing the red meat allergy, and we have not entirely ruled out the possibility that there may be some virus or pathogen that's co-committing or co-transmitting rather with these ticks that are developing into AGS. There's also some evidence. There was recently a study in the state of Maine suggesting that, ticks other than Lone Star ticks can also lead to the alpha-gal syndrome. So much still to be understood, and much to be understood from assessing risk in the proper way, not just looking at where the ticks are and who's getting bit by ticks, but sort of understanding qualitatively how these tick bites are leading to disease. Oh, here's the slide that I meant to show you before. This just tells you the incidence of these different pathogens among the ticks. So this is a tail of those ones that I've been telling you about. So what I want to point out to you is that Borrelia burgdorferi is absent from Lone Star ticks and dog ticks. We've tested on the order of 80,000 ticks in New England over the course of couple decades, and never find those pathogens. So the notion that dog ticks or Lone Star ticks are transmitting Lyme disease just isn't the case. Things like Babesia and Anaplasma, 5 to 10%. There's a range there depending on different parts of the different geographic regions. So if we got rid of the one on the top, Borrelia burgdorferi, we'd still have a problem with 5 to 10% of the human biting black-legged ticks. I mentioned to you before that American dog ticks are very rarely infected, and you see some of the numbers there. So too, Lone Star ticks have a lower infection rate with the different Ehrlichias. So it's these guys on the top for today anyway that are the primary vectors of disease. But with the other ones moving in and increasing in number, we expect that they may be bigger players also in the future. That's just summarizing what I said before, that understanding tick exposure is understanding the tick species, the infection status, and the duration feeding. Three components of exposure risk that are helpful in making that consideration. So I'm going to just show you a quick little animation here. This is precisely why. So if you have questions about why it is that ticks have to feed for a while in order for pathogens to move out, it basically has to do with the physiology of the tick. The blood comes in through the hypostome into that organ on the left, which is the midgut of the tick. And in order to get out of the tick, it has to actually migrate over to the green balls on the side, which are the salivary glands, which means it has to navigate through the open circulatory system of the ticks. It doesn't even have the ability to do that without the next blood meal that brings in serine proteases that allow it to cut through the membranes and make it across. And all of that takes time. And so it's a real biological process. And I emphasize again and again that ticks aren't syringes that suck up and spit out. When they suck up, they have to undergo a process of digestion in order to move the pathogens into the area where they can come out of that tick. And this takes on average 24 to 48 hours. It just shows the flow of blood versus saliva. And how do we use that to assess risk? Well, we can look at tick bodies, and you can train your eye to understand based on the change in the tick's body shape, or you can have an expert do it for you. You can look at the change in the body shape of the nymphal tick or the adult tick. And here, again, remember, ticks can feed up to 7 days. As they get bigger, it means they've fed longer, and you know that when they've crossed that threshold that they're likely to have transmitted if they were infected. Sorry, that's just a repeat tick. So how can you figure that out? You can go to places like tickencounter.org. That's our colleagues at the University of Rhode Island. They have a free service that they you can send them a picture of a tick, and they can tell you something about the species. They can tell you something about how long the tick is fed and give you some assessment of risk. They can't tell you whether it's infected or not. You can go to one of the tick testing sites like ours at tickreport.com, and there they can go do a thorough workup to tell you whether the tick is infected with a particular pathogen if you needed to assess its risk. So just in closing, I just want to say that something about the center that we started here. So in 2022, we started the Center of the New England Center of Excellence in Vector-Borne Diseases, or NUVEC. Our logo's down in the right there. And we were set up to do three things. So it was a $10 million grant from CDC, came to my lab. We partnered with academic laboratories in each of the New England states to conduct operational research. That's research that really makes a difference in figuring out how to reduce the number of people getting bitten by ticks. We're training the next generation of medical entomologists, and then we're building these communities of practice, or we're trying to figure out ways to build communities of practice. This was one of the charges from the CDC, was to build communities of practice. And as I see it, school nurses are maybe potentially good partners in this community of practice, because as I understand it, because controlling ticks is so much different than controlling mosquitoes, we really have to rely on not agencies that are going to come in. There's no, I don't imagine there's going to be an agency that comes in and I'm snickering because I never thought there would be something called Space Force either, but I don't imagine there's ever going to be a body called the tick eradication state or federal agencies to do it. It really falls on individual residents to maybe do something about ticks in their backyard, and or each of us to do something to protect ourselves and our loved ones and our pets from bites. And so in that regard, this community of practice notion is that there are people that are making decisions about what they want to do, the deciders. There are providers that are providing good materials like some of the acaricides and the repellents and the specialized clothing. And then there are guiders. There are people that are trying to tell the deciders what's good from those providers. And I think in this case, this is where people like school nurses, we're also thinking librarians can be part of this sort of maybe a little bit of a non-traditional public health role for librarians, but on Cape Cod, the librarians have sort of embraced this idea. And so we're trying to develop these notions of how we can build these networks and make sure that people understand, have all the information they need to make the decisions they make about how they're going to assess risk and mitigate risk of tick-borne diseases. This is just an example of what those community practice. So you can imagine deciders might be hikers. Someone's going for a hike, they got to make a decision. There are outdoor goods stores that provide lots of different options for them to take on the trail with them. And land managers and departments of conservation and resources could be the guiders to tell them, well, this is something we think that works very effectively. This is cost effective. This has this, there are consequences of this approach, et cetera, et cetera. So we look forward as a center to working with groups that want to help us to build that community of practice. So in closing, what's the post-Lyme world? Well, ticks will still be part of the landscape. They're not going away. If they go away, it will be very much by accident and will have probably introduced a worse problem. There are other pathogens that can be affected, even if we were eradicate Lyme, which is not likely to happen in the very immediate future. And that learning about basic biology of ticks and tick-borne diseases remains a fundamental aim. There are something like, and we recently, we realized that no one had actually estimated it. So we just went and did a back of the envelope estimation of the number of people that get bitten by ticks. And we think in New England, about 1.6 million people minimally get bit by ticks every year, given that about 400,000 of them result in Lyme disease. So we think that this is a principal concern and something that people should understand better and be able to do something to try to stop. So I'll close by just encouraging you this little piece of advice to be tick safe.
That got a great chuckle. Thank you so much. We do have a few minutes for questions. Anybody, you have a question here?
So, and they're recommended, if they're on for 24 to 48 hours, different providers will say different things about in the doxy for the 24-hour dose or not treating and just monitoring.
Yeah.
Practice.
Could you hear that, Stephen, or would you like me to repeat it? So the question was about treatment and whether there should be different treatment based on 24 hours versus the 48 hours that the tick was likely on the patient. Yeah. Or just monitoring.
Just monitoring. We always emphasize that these risk assessments are not meant to take the place of diagnosis. So yeah, I don't think there's a difference between 24 and 48 hours. It's just more about understanding the risk. Was there a question also about the 2-dose prophylaxis, or was I just reading that into it?
Is that the gold standard, that prophylaxis?
No, in fact, there's only one study that's maybe two now that have actually talked about this. It is a common practice to use a 2-dose prophylaxis. It's not altogether clear that that's effective. The typical therapy is a 2 to 3 week course with doxy or amoxicillin if doxycycline's counterindicated.
Other questions? I actually have a couple myself. So I had a practice when I was a school nurse of if we found a tick that was attached to a student, we would remove it immediately, but we would tape it to a piece of paper with the date and the time and the location and the student's name and send that home with the student so that if the student were to develop symptoms, the parent could bring that piece of paper to the doctor, and that would help identify the type of tick or maybe how engorged it was, how long. Is there any part of that that's part of the protocol, or was that just us being neurotic? What would you recommend?
No, I think that's a great idea. I would say that doctors, MDs are not particularly good at identifying tick species. We've shown this again and again. And so they may misidentify them, and they're not very adept at telling the difference between how long the tick has been on. Tick Encounter Resource Center, the one I showed at URI, They are very effective at doing that, and they can guide the doctor and the individual. That's a very good practice. Keep the tick, because we say that tick is a record of that exposure. So weeks down the line, febrile illness and, sweats and all this other stuff, you can go back and you can assess whether that tick is the right species and or has fed and make those determinations. The other thing that we often tell people to do is to think about putting just a Sharpie mark at the spot that they took the tick off, so that they remember, oh yeah, that's where I had the tick. And so if you get the bullseye rash or get one of those kind of indications, you can localize it to the place where you had a tick bite before.
Thank you.
What about sending the tick out to be tested? Some families will ask if they want to send it out.
The question is, families will ask the school nurse about sending the tick out to be tested. Are there resources on that? What's the recommendation?
There are. So UNH started a tick testing program recently. We tested ticks for them, and now I think they're starting to do it themselves. They are basically co-opting in a COVID lab to do tick testing. University of Maine does it, but I think it's only for Maine residents. And so too the University of Connecticut, the Connecticut Agricultural Experiment Station. And then there are laboratories, fee-for-service laboratories that will test ticks as well. Yeah, so there are a bunch of different options now for where you can get ticks tested if that's desirable. But most of them are either state-specific and or associated with some kind of fees.
Other questions? Yes, in the back.
Maybe recommended only if symptoms are presented in the student first.
Did you hear that? Sending the ticks out to be tested. Is that, do you wait until there are symptoms, or do you just send it once you have the tick?
So my opinion is that the best use of the tick testing is to get it in advance of the symptoms, in advance of serology. So obviously a serology is going to take 2 to 3 weeks after the exposure before that would manifest. The idea of tick testing is to get the risk assessed earlier on and to maybe take some actions as you're leading up to symptoms. If symptoms are in evidence, then for the most part, probably the serology is going to derive what that symptomology is associated with, and you're better off just going that way. And it's probably too late, unless it's something extreme like, I mentioned Rocky Mountain spotted fever. We have seen cases of Rocky Mountain spotted fever in the upper Midwest that where the tick test was helpful because it was something that docs weren't looking for. There were 2 cases of tularemia that kind of went that same way. But for conventional Lyme, like if you've got a serology, you've got symptoms, the tick test isn't really going to help you. The treatment. Every indication is that so everyone hears the story about long-term Lyme and chronic Lyme. Every indication is that the earlier it's caught and treated, the more likely there's a favorable outcome.
You're gonna have to put out about chronic Lyme versus once you get treated, people still feel like you have chronic Lyme,
Yeah.
start from that.
What can you tell us about the incidence of those that will go on to develop that chronic Lyme? Is that frequent, or what's the data show?
That's the third rail here. So there are lots of folks that are suffering from long-term consequences of whether it's all attributable to tick exposure and or Borrelia burgdorferi remains an open question. But I think the pretty good numbers are that about 80% of people will respond very well to antibiotics and not experience consequences after that.
Yeah, in the back.
Post removal of a tick, is there anything you can do? You know how if you get a bee sting, you can use a meat tenderizer to break down the protein. Is there anything you recommend that you treat the spot after removal, or don't treat the spot?
Right. Recommendations post removing a tick. Is there any treatment for that spot to, I guess, either decrease risk or decrease symptoms? She mentioned that, like with a bee sting, you can use a meat tenderizer. Is that what you said? To neutralize proteins. So that was. Is there any recommendation of what to do with the site after a tick is removed?
Topical antibiotics, you rub that on the side of the wound. There's I thought I heard a little bit of the question. There's a device out there now called a bug bite thing. Have people heard about this? So
Yeah.
it's basically a little suction device, and people swear that it relieves pain of bee stings and mosquito bites. I haven't heard it associated with tick bites, but it's it's another thing that's out there. I think topical treatment's a good idea.
Right. Thank you.
It's a wound, right? So anything that you would do to a wound.
Yeah. Yeah. Awesome. Is there another hand? Yep. One last question for you. Thank you so much for your time today. Tick tubes. There was an initiative in a community that I heard of where it was almost like a community effort. They had the supplies out and people came and made their own tick tubes to bring home to their yards, etc. I just didn't hear you mention that. Is that an effective thing to do even?
Yeah. So the tick tubes are like a toilet paper roll that you put a cotton inside, and the cotton is treated with permethrin. And the notion is that white-footed mice, which feed a lot of the juvenile ticks, will take the cotton and then they'll line their nests with it, and then the acaricide will kill the ticks in the nests. There's been something on the order of like 6 different studies now. Most of them suggest that it's not very effective. A couple of them have suggested it's marginally effective. One of the reasons that it may be effective some places and not others is that if there's chipmunks, then the chipmunks don't use the tubes and you get Lyme disease on the chipmunks and it just goes on without. Probably has also to do with the density of the ticks and different things like that. Lots of ecology. So like in many things, tick control wise, it's like it's ecology. So sometimes things work and sometimes they don't. And the number of times that I told people that something doesn't work and then they come back and tell me, well, it worked in my yard. I'm reminded that people's lived experience is a reality.
That's right. Well, thank you so much for such an informative presentation and great advice, as always.
Right here.
Thank you so much and have a great rest of your day.
Take care. Thank you.
Bye-bye.